So in this video, we're moving on to the part where we actually take the sequences that we found and try to draw some trees with them. But the first thing that we do is do some sort of alignment. And in this video specifically, we're going to utilize the system of UGENE as our graphical placement for this. So if we go to UGENE, you can see that we have a plasmid that we looked at before. And so what we're going to do now is bring our files into this, right? We don't really need this one, but we can leave it open. I'm just going to go to the apes, and I'm going to pick on all of these, right? So let's see how well this works, because this is not a huge amount of data. As you can see there, usually the largest one is like 230 kilobytes, so it's not going to be that bad. So here we go. And it's basically trying to see which format it is. It's plain text, the Denisova GB. Let us see. Yeah, let us see what happens when we try to open them. It should try to realize what they are. And we had no problem with GenBank files before, but here they are. Oh, some of these are not loading properly. And let's see why that is. So why did we get only one thing opening up properly? I do not know. So let's just remove some of these things. It also might have something to do with the way that I downloaded them, where they're not appropriate. So once again, the beauty of plain text is that you can try to open these things up in your text editor or in your thing here, right? So we're going to go, oops, we're going to go there. And we're going to look at do we have one with an H? Homo sapien. Yeah. So yes, as I suspected, they actually did not download properly, right? So that is totally fine, right? So we're going to go and re-download these sequences appropriately. These came from, had these in GitHub, so we'll go there. And I thought I had done it appropriately, but I had not. So, and that's exactly why it did not open properly. So they're probably under data. You would think they're under sequences, but they're not. Under barcode and under alignment. And so, nope. These are actually the homework ones. So let's see. Data. Maybe they are under sequences then. Sequences. Barcoding. There we go. Oh, there's ape. Should go back up. Okay, ape. And so the gorilla, let's just open it up. If we open these up, it'll, and we can download them once again. Right, so yeah, so here's the raw thing, and I can actually just take that. That's what happened. It basically gave me the GitHub page HTML as opposed to the actual raw sequence. So that also goes to show you that if you come across a problem, be able to solve that problem. Especially when you're dealing with computational things and different file formats and different repositories. So now they all look like GenBank files like we're used to. So we'll just save these locally, and they should replace everything that we have. And then we'll actually go over why it's important to have these annotations in the GenBank files as opposed to the FastA files. Because at the end of the day, there are useful features that we might want to extract to make our computational analysis easier on us, right? Like I said, we don't need to look at the whole mitochondrial genome, we can deal with things in another way by looking at specific genes. So you can see here even, this is the annotated version of the pygmy marmoset one, the Calithrix, and you can see that it's actually labeled as Calithrix, but here you can see the genome is labeled by its accession number. And so let's go back and try to, once again, bring in our various sequences here. Helophates, Pileatus. So some of these actually did not, I think they all kind of failed in some way. So symphylic, and then that's it. Let's try to see if they open up now. So now this is what I was expecting. So it realizes that there are multiple sequences. You can separate the sequences and they'll just be separate entries, such as we see here. We have 2 entries on the side. Or you can merge them into some sort of alignment or a multifast A. We're just going to keep them as separate sequences for now. And here you can see them loading. And it's done, right? So you can see some of these are fast A, so there's no annotated map. We can go and look between these tabs to see the different things. And notice that this one is the gorilla one. And if we look at it, it is greater than 16,000, almost 17,000. So for some reason, this gorilla one, right, we can actually do, we can actually select the sequence and mark it as circular. And then we'll see the genome in the appropriate view, right? So that's more what we're expecting. Calithrix and so forth, right? So I'm just trying to make sure, because it doesn't seem like everything loaded. Did this one load? Yes. Peniscus, Symphilingus, Pongro abeli, Neanderthal, right? Lemur. The Denisovin is not here, so let's see why that is. No, so there's something wrong with the Denisovin file as well. And that one is kind of interesting, so I'm going to try to see, do we have it here? Yeah. So once again, let's just open that one and get the raw version of it. Denisova. Place that one. Yeah. So now that we've done that, we should be able to import it properly because it's not just HTML, but the actual file itself. Yeah, there we go. So as you can see, we have all these different files. They have their names. They're kind of useful that way. And yep, so that's all those things. You can see the annotations of these, and you can see that sometimes they have 2 annotations, right? Sometimes they'll be here like something that's an actual gene-like name, site B, and then you can see here that's an accession number that you can find on NCBI, right? Let's see this one. Yeah, this one also does the same thing. So you can see that they're all about 16,000 and a half, so 16.5 kilobase. Let's go back to the marmoset. Yeah, even the marmoset, the monkey, and the lemur, right? The lemur is 17,000. So the lemur has a larger one, like by 500 base pairs. In any case, it's kind of irrelevant, but you at least notice that these annotations are very useful because they tell you the same genes. Typically, when we do barcoding, we are going to use like cytochrome oxidase 1, sometimes called COI, sometimes called COX1. Other times, you're going to use something called cytochrome B. Sometimes you'll use, you know, a portion of various genes, you know, but that's what we're looking at here. So let's think about doing this alignment, right? So this is how we've imported our sequences here, and we're going to now just systematically select these guys. I'm basically just selecting them using the control button and clicking on them because I'm on a Linux system. If you're on a Mac, of course you use the command key probably, and that will work out. And so I've selected these, and I will right click or command click, and I'm going to actually export the sequences as an alignment, right? And so basically, I'm just going to do the apes alignment. And it's going to ask us for a format file of the file format to use. I'm fine using this Clustal W. Sometimes you'll use these other formats such as Nexus, which has a lot of information in it. It's a very current format, or the Philip versions. I'm fine just doing Clustal for now because it's not that urgent for us to understand what these things are. So we go and here it is. Now what we've done here, you can see, is it just basically put them all into one view, right? And here they are all as one view. And this is why I was very picky about moving my names around, because some of these names are not useful for us to go by, right? So let's edit the sequence name, and when we do that, it'll become easier for us to read, right? So I'm just gonna rename some of these now, just to show you that it can be done. Because a lot of times people just do what is minimally asked of them, and don't necessarily go on to make it readable. And sometimes making things readable and presentable also makes things more professional in nature, right? So here we go. So once again, little bits of housekeeping make it just better at the end for us. It can be tedious, but also very useful nonetheless. And even this one, I should rename it as Homo sapiens neanderthalensis, which might even be wrong. Neanderthalensis, right? As best as I can. This one, I don't know what it is. FR69. So that's a Denisovan, as I can see right here. So edit that. Homo. Right. And let's go to this one, HM0213. So that's the Panpaniscus. So we can just cheat and write common chimp, right? We're just gonna have common names. It doesn't make so much of a difference here. Let's see, 1643. That's this one. So that's the bonobo. So that's the pygmy chimp, or the bonobo. 1646 is the Pongo pygmaeus. Now I don't know the common name for that one, but that's an orangutan, so we're just gonna call it Pongo pygmaeus. NC 1112. That's the gorilla. So remember there were two species, but I only have the one. So we'll just call it gorilla because I don't remember the common name for this type of gorilla. 249, and that's the human. So we'll just cheat and just call it human. It's a little unfair to the other humans that are on here, but you know, these are the ones that exist, and this is one that exists, right? It's a representative one. It's not a consensus or anything like that. 22776. So I don't know this lemur's common name, but 2776 is lar. And then 45 is pilatus, pileatus. And 42 is that's the pygmy chimp, pygmy marmoset. So we're just gonna write pygmy marmoset, right? And then what's this AJ45? AJ45. So that's this lemur. Okay, so that's the other outgroup. That's the other negative control, right? So lemur, what is it, kata, right? Anyway, so what we have here is it's a little bit easier for us to parse through, and we can go and see how there's no rhyme or reason. They just have these colorations, and once again, you can see these dashes for gaps. So we remember the lemur had the longest one. And what we're going to do now is right click or command click on this area, and we're going to do an alignment, and we're going to use align with muscle. Muscle is one specific type of multiple sequence alignment algorithm. As you can see, they offer other ones. We're just going to use that one, and once again, because we're not getting into details of how these things work so much, we're going to do the default ways of doing it, right? So we'll just click on align, and you can see that it's computationally doing something, and it's a little bit stalled at 49%. So we'll wait. So remember, these files are not huge, but obviously what's happening is the algorithm is doing a lot of computations to try to match them against each other, and we will see the output of that soon because it jumped from 49 to 74%, and it will, incidentally, you can see it made this alignment file because we called it apes, and that's what this is. Okay, so we're back now, and what happened was it took a very long time. It was very computationally intensive. Even though these files are very small, it's doing something very heavy in terms of doing its alignment. And what you see here is a bunch of dashes, but what that ultimately means is that it has aligned them to the best of their ability. And so you can see down here, there's a sort of like a graphical representation of the best alignments, right, that have occurred. So the highest alignments are like those that reach all the way to the top. And if we scroll along here, we can see what has happened. It introduced a bunch of gaps. You see some colors no longer match up, but the context around these areas is preserved. Right, and that's what we mean by having an alignment and looking at these things, right? So we can scroll along and you see how they mostly match up, and where they don't, there might be a gap or whatnot. So we can scroll along and see what has happened. We remember the longest one was the lemur, and you can see how it just sort of like is still up there at this point, with inserting a gap for everything else and whatnot that we see on here. So we have created an alignment, and it took a very long time and was computationally intensive, right? There's a shortcut for this, and as I said before, we often use a barcode such as cytochrome oxidase, which is a much smaller subsection of this. You can use the whole gene or part of it, as long as it preserves enough information in the context that we're looking at to do these types of comparisons against each other. So now that we've done this, what we're going to do is click on this icon here, which is to create a tree. And so we're going to click on it, and it gives you this menu panel, and we're going to build a tree, right? And this is when we have several choices, and we're going to do this multiple times to look at the differences of these things. And as I said, if we were going to not use a very computationally intensive algorithm, we would do something using distance measurements. And so what we don't see here is that there's a scoring system that has been made for these alignments. And so what we will do now is you can see right now the tree building method. There's a drop down. There's something called Mr. Bayes, which uses Bayesian statistics and inference, right? That is a nice evolutionary model system because it takes into account what we call priors. Things that happened earlier are predictive of things that will happen later, and that makes total sense. There's something called PhiML, which is and ML stands for maximum likelihood. That's another statistical methodology of looking at these relationships. And then here is phylip neighbor joining. Neighbor joining is very computationally inexpensive, right? And so we're going to try that first. This is not necessarily a good one to use for evolutionary relatedness, right? So we're just going to show you because this will happen very quickly because it is inexpensive computationally. There are some other things that are used here. You can choose different types of distance matrix models, and that influences the types of statistics it's using to actually do these calculations, because there are some involved. We're just going to use the default F84, and you can also see here something called bootstrapping consensus tree. You can enable bootstrapping, we're not, because that will increase the amount of the way that it's trying to build a tree, and increase the amount of computations, right? Display options, we can display in the alignment editor or in a new window. We'll do it in a new window. Distance matrix is fine. Bootstrapping, we'll talk about some of those things. And it's going to save this information in our directory system here. And it's going to create what we call a Newick file, NWK. Now you can go to save settings, and save settings are not really bringing anything up, so that is problematic. But essentially, the save settings are what we chose, sorry, as the default, sorry. And so that's fine, and what we'll do is build the tree now, and it should not take very long. And so you can see, once again, the progress bar is pretty much done, and here it is, right? So we have this new file, and here we are. Right? So we have a very basic tree. And as I said, this is not necessarily the best way to do it. However, if you look at our outgroups, the outgroups are outside of everything else. So here's the monkey, here's the lemur, and everything else is over here, right? That's the inexpensive way to do it. Not the best way, but if you want a ballpark figure to look at, here it is. And so let's go back to our alignment, right? So here's our alignment, and once again we can build a tree. And now what we're going to do is choose maximum likelihood. Now notice that the options have changed here, right? Maximum likelihood, once again, we use what's called the substitution model, and the substitution model has all of these options, right? So I'm prone to using this default one called HKY85. It's from a paper published in 1985, and essentially what it takes into account is what we call an evolutionary model. And as you see here, there's something called a transition, and there's something called a transversion. And that's when we need to talk about basic molecular biology once again, because that'll help us understand what these things are in terms of the types of model that we're talking about, right? So here's our whiteboard once again, and we're going to choose a different page size because this is not appropriate. And so here we are with our whiteboard, and we can talk about the things that were mentioned, right? So we had things that were called transitions and transversions. And we know that we have purions and we have pyrimidions, right? So transitions are the things that are easier to occur, right? In terms of what we have chemically involved here, right? So transitions versus transversion, and there's a standard diagram that we often see, and it very accurately illustrates to us what a transition and a transversion would be. So transition is simpler than a transversion, right? And if we look at our nucleotides, the purines and the pyrimidions, the major difference is that the purines are larger. They have 2 rings associated with them, right? I'm not drawing them properly because there are various nitrogens involved and double bonds, whereas the pyrimidines are single ring structures, right? And so basically, we understand that it's easy for a purine to basically have a transition to another purine. And pyrimidine can change easier to another pyrimidine, right? So this is something that chemically can happen because of various chemical changes from exposure to toxins, exposure to other chemicals, radiation, and things like that, where these types of changes are understandably so, right? A single ring to a single ring, a double ring to a double ring. And so these are what we call the transitions, right? So that's what these are. Oops. And there's no extra points for penmanship or coordination here. But, however, if we have any sort of purine or a pyrimidine, this is the transversion. It's basically converted from one type of molecule to another, right? A double ring to a single ring, or single ring to a double ring, right? And that will alter things. So a transversion is more difficult to occur when you look at it chemically. So there's a way that we rate that sort of change, right? We rate that sort of change differently because it's more difficult to occur, right? So pixels. Okay, so the standard diagram that you will often see is that we have A, G, and C, and T, right? And we can basically draw some lines here, and we know that we already said that this sort of change is fairly easy, as is this type of change, right? So we said that this was transition, right? This is also a transition. And the other thing that you see is that's more difficult is here's a purine becoming a pyrimidine, or pyrimidine becoming a purine. Same here, right? So this is more difficult. And this is also a transversion. And this is also a transversion, right? So those are the transversions, right? So that is how we understand these differences, right? We know that there is a difference, that it's chemically easier for a single ring to become a single ring, a double ring to become a double ring of the other type. And those are expected mutations that occur, and they have a different weight, as you can understand, right? So when we go back to our UGENE, right, we have to understand that HKY85 takes into account the likelihood of these changes to occur over time, right? The likelihood of mistakes to happen, the likelihood of other types of mutations to occur that would bring about these changes, right? And also things that would insert, spaces or delete spaces between different nucleotides. Remember, not everything is a gene. Even in a mitochondrion, which is a very small genome, not everything is a gene. So having a gap or a deletion or a change isn't going to really change the function of that genome so much, right? It's when it's in the genes and the coding sequences are going to be altered that we have these issues, right? So you can modify some of these things. So we have estimate instead of fix, so we just leave that box clicked off. Proportion of invariable sites. And so once again, we're not going to get into all of these fine details. So we're just going to say that's maximum likelihood just to make it easy. Branch support, these are also different versions of branch support. We can perform a bootstrap. You can increase the number of bootstraps, and so otherwise it defaults to something. And so bootstrap is basically how it's matching up these scores of these sequences in the alignment, and actually trying out how well they fit in a certain location on a tree. And it's trying it out multiple times. And that's what a bootstrap is, to see the correctness of that fit, right? And so it'll grow a leaf on it, and it'll prune a leaf, statistically speaking, right? And so there's like all these things. You can also train it a little, right, against other trees and other things that can happen here. And so we just leave these as default because we don't need to discuss how these things are working, because this is molecular biology and not hardcore sort of bioinformatics in terms of understanding how these work. It also requires a lot of statistical knowledge. So let's press build, and this will take longer than what happened before, right? So you see it's a little bit slower, whereas neighbor joining just popped up right away. So this is going to possibly take a little bit more time, and will maybe require another pause in the recording in order to ameliorate the load of time and resources here. So as we can see, the alignment and tree building using maximum likelihood finished. It took quite a bit of time to do so, as opposed to the neighbor joining, which was almost instantaneous. So we can see this here. We can actually change some of these functions on here to make it a little bit more presentable. So we're going to click on this dendrogram thing here, and that gives you the options for the tree settings. One strange thing about UGENE is it defaults to this gray, which is kind of hard to look at. So you can just go the font settings and change it to black. It makes it easier to see. Yes, much better. And also you can change things with respect to the other pen issues. Fortunately, other things are already in black, like these lines. You can change the thickness of things. The line width can be increased, right? And essentially, what you do see here are some numbers, once again, and we saw that also with the neighbor joining tree, right? So this is the neighbor joining one. Once again, let's change the coloration just to make it better. And we can see that you know, we have a difference in the appearance of these. You can change the fonts, and more importantly, what you can do is look at it in this way, and you can see once again the lemur and the pygmy marmoset are the appropriate outgroups. They're not part of what we see here. Now, one thing you can do is toggle some of these things. I traditionally would like to align these labels so that you know, they're all at the end, and this will make more sense when we alter the way that this figure looks, right? You can show the distances, and now you see a lot of things here, and you can actually look at this as a phylogram. Now this is when you're going to see those alignments of labels is important, right? So now we can see that the branch lengths are actually informing us of a lot more than what we knew before, right? So aligning the labels out here is really important because, as you can see, these branch lengths are quite different now. I'm going to actually collapse some of these things just because it's a little bit messy. So yeah, so let's use the distances. Sometimes you can have the bootstrap scores written here, which some of it seems to be, and you can actually have this understanding here of, here we have 3 types of humans: Homo sapiens, sapiens, the modern human, the Neanderthals, and the Denisovan, right? And you can see how even here with the 3 humans, they are clustered together properly, as you would expect. And they are somewhat related. And then, of course, you have adjacent to it the 2 types of chimpanzees, the bonobo, the pygmy chimpanzee, and the common chimp, and they are the most closely related there, based on this mitochondrial genome in its entirety, right? The gorilla is then a little bit further off, right? And then a little further off are the 2 orangutans. Notice that the orangutans are on the same location, right? Same branch, so that makes a lot of sense given their similarity. Here we have the 2 lemurs of the Hylobates genus clustered together. The one Symphalangus is off, and then the one Damascus that we had chosen is one off, right? And then you can see how we have these are the great apes over here, and then these are the lesser apes, and then we have the monkeys and the lemurs over there, right? So in any case, what is the utility of this? Well, like I said, neighbor joining is great if you just want to see something very quickly. You can do it this way as well, and also have something similar displayed to us, right? And the other thing that we have before us is that, remember, we save these files as Newick files. They're in our file system. So now what you can do is, these aren't necessarily the easiest things to look at, but you can see some differences here, right? So here we have the chimps, the humans, the orangutans, and we have the lemurs, not the lemurs, the gibbons, but they're a little bit swapped in the way that they are branching off here. And we can definitely see the monkey and the lemur are in the distance. Now, what you can do is do some other types of manipulations subsequently. And that's where these Newick files that are in our file system are very useful. So at this time, I will now launch another, interesting. Just a second. What we can do is go and launch another piece of software, and let's see if we have it installed, because it was supposed to have been installed before. So I'm searching for this piece of software, and let's see, it is already the newest one. Okay, so for some reason I can't find it, so we'll just do it this way. At this moment in the background, I'm waiting for the software to load properly. And here it is. This software is called FigTree. And FigTree is great for helping us manipulate our trees so that we can view them better. So here we go. Let's open up one of our tree files. And basically, we have to find it. It's in our home directory, wherever that may be. There's my home directory. It's in the sequences file in the apes. And we're going to open up the maximum likelihood tree, right? So here we go. Here's our maximum likelihood tree. It is showing up here. Now, I'm going to drop down into the terminal once again here, and we're going to look at what one of these Newick files look like. Here we're going to go open that up. And first, let's look at the alignment file, right? So the alignment file in this Clustal format is simply a textual thing once again, and it shows basically something similar to what we see in the output that's more colorful in UGENE, right? The other thing that we can do is look at the other file, which is the Newick file. We'll look at the maximum likelihood one, and that is also a text file, as we can see here. And so the Newick file is very useful for us to use as a textual file that we can pass on to each other, because as you can see, what it does is it has a certain syntax to it of open parentheses that illustrate to us what a branch is, and commas and all these other types of annotations. So these are always very useful for us to study, and also to modify in a way that things will be displayed properly later on, right? So as I said, we can actually modify it a little. So for example, let's say that I wanted to modify it. We can do this in a text editor. I'm just going to do it here because we're already looking at it. And basically, I have everything said Homo sapiens, except for human, right? The modern human. So we'll just say Homo sapiens sapiens, and I'm just going to go scroll over to that area where I can do the appropriate sapiens, the appropriate modification, and there we go, right? So I have made that modification, and notice that FigTree realizes that it was changed, so we're just going to change it. Yes to all. And basically, oh, this is actually in UGENE that's seeing this change. So fine. So let's go away. Yeah, this is now it's just complaining. So this is just FigTree. We'll just ignore that in UGENE. So we'll go back to FigTree, and we can look at it, and it did not change it here. Right, that was the UGENE that recognized it, and so we can't really read any of this. So once again, we can play with our tools where we can increase the font sizes. Hey, now it's readable, right? Now it's completely legible. We can go to have the node labels if we want to. We can actually have the branch labels if we wanted to. And we can also toggle this so that we can change their font sizes as well to become more prominent. This is like extremely useful. It's a little bit more flexible than what we had seen built into UGENE. And what we can do is also add a scale axis if we wanted to. So now it's telling us about various issues of distance of time, and we can align the tips as we did before. We can actually change some of the appearance with the coloration. The line weight we can actually increase, right? So that looks a little bit better. It's more prominent. We can do other things to make it a little bit different, such as changing the curvature. That really serves no function other than the fact that you might prefer the way it looks. And you can do other types of modifications to this. Another thing that's useful is, you can actually change these types of roots that we have here, right? So if we look at this, let's say we have the pygmy marmoset. Pygmy marmoset is a little bit closer to this ape line than the lemurs. The lemurs had diverged such a long time ago from the other things. But what you can do is do some annotations. You can reroute things, right? So now it's all rerouted to the pygmy marmoset. And now you see how the branches have changed. We probably don't want that so much, but what we can do is maybe click lemur and reroute that as well, right? Now you can see how it's switching things. And once again, this is not necessarily the best option for this, but what you can do additionally is you can think about these things. Let's reroute that this way. No, one branch must be done. That's fine. You can also have other views such as this. It's not necessarily that useful, but sometimes it is in with respect to visualizing something that's very complicated. You can also change it this way, where it's not rooted anymore, right? Yeah, let's go back here, and what we can do is essentially we can change some aspects of this, and like we can say here that we can highlight. We can highlight the lesser apes, right? So let's select a bunch of these here. So I'm just holding on to. Well, we could just do it like this probably. Um, and so these are our gibbons. So we can highlight this, right? And maybe the gibbons are some sort of orangey thing going on there, right? Yeah. So those are our gibbons, right? And then maybe we want to talk about this cluster of organisms, the gorillas and the bonobos and the chimps and the humans, and let's highlight them as well, and let's make them like a bright blue color perhaps, right? So, and then for some reason it didn't include the gorilla. I don't know why. But in any case, you can see how we have these things highlighted here. They might be meaningful, they may not be. But I'm trying to inform you that you have the option and flexibility to do many sorts of visualizations and annotations of these. And, all of these things that we can think about changing. And otherwise, what you can also do is export them to various graphical standards. So I'm going to export this as an SVG because that is a very useful sort of file format for dealing with curved lines and text and colorations, so that things can be stretched out and read properly. So that's the very useful thing that we can do with these sort of software that we have in front of us, right? So yeah, so what we're going to do now is move along and talk about other aspects of these types of exercises. And that means not necessarily doing everything in UGENE, but more command line based things.